And first of all, I'd like to thank the organizers and Sophion people for giving me this opportunity. Today, I'd like to talk about the roles of sodium channel clustering at the axon initial segment of neurons and its regulation by cytoskeletons, particularly focusing on our findings in the auditory system. So the axon initial segment is a highly specialized neuronal subregion that separates axonal and somatodendritic compartments and involved in initiation of action potentials. This is because this region has high density of voltage-gated sodium channels and shows the lowest threshold for action potentials. This is a schematic drawing of molecular organization of the ARS, and you can see many kinds of proteins are localized at the ARS, such as cytoskeletons and scaffold, cell adhesion molecule, and ion channels. All these form huge protein complex, and it is characterized by a periodic arrangement of actin rings separated by spectrin tetramers at an interval of about 190 nanometers. Importantly, sodium channels are tethered to this actin spectrin meshwork via their scaffold ankyrin G, which enables these channels to localize at this site. So it is now evident the distribution of the ARS is more diverse than previously thought and differs among neuronal types and brain regions. For example, the ARS is very long in layer 5 pyramidal neuron, while it is much shorter in cortical interneurons and cerebellar Purkinje neurons. Since the ARS is the site of action potential generation, these structural characteristics of the ARS should have strong impact on the membrane excitability of neurons. This is a computational model showing the effects of length and distance of the ARS on threshold current required for generating an action potentials. And you can see the red area that represents small threshold current and the highest membrane excitability shows a negative correlation between the parameters. We have studied the functional implication of this AS distribution in nucleus magnocellaris of the chicken, which is a homologue of mammalian anteroventral cochlear nucleus and involved in precise relay of auditory signals for binaural spatial hearing. NM is organized tonotopically such that neurons tuned to high-frequency sound are located rostromedially, while those to low-frequency sound are located caudolaterally. We have shown that NM neurons differ in the length of the ARS along this tonotopic axis, with the length becoming shorter as the tuning frequency of neurons becomes higher. This is an adaptation to the patterns of synaptic inputs at each frequency. So that high-frequency cells receive a single large input from an end-valve terminal, and this preserves the timing of presynaptic spikes with high-fidelity transmission. On the other hand, low-frequency cells receive multiple button terminals, and this convergence of input reduces the jitter in the postsynaptic spiking by eliminating the effects of temporally displaced input, and this improves the quality of temporal coding. However, the convergence of inputs reduces the excitability of neurons because it slows the rise of synaptic potentials and causes sodium current inactivation and potassium current activation. Therefore, the long ARS is particularly important for the low-frequency cells to overcome this reduction of excitability during the converging input. On the other hand, the short ARS in high-frequency cells can prevent aberrant spikes during this large input, and it is suitable for high-fidelity transmission. Thus, the AS distribution is effectively coupled with patterns of synaptic inputs in NM, ensuring precise and reliable temporal coding in this nucleus. Importantly, these structural characteristics of the AIS are not fixed, but can be altered dynamically by neuronal activity, implying that the AIS is a site of plasticity. This is an example of AIS plasticity in NM. So we here deprived afferent input, auditory input, by removing cochlea mechanically, and this elongated AIS and increased membrane excitability and caused spontaneous firing in NM neurons. Neural activity is important for survival neurons because it facilitates release of trophic factors. So we consider that the elongation of AIS in NM neurons would maintain the integrity of auditory circuit by restoring activity after hearing loss. Recently, we reported another role of AIS plasticity, which is to shape the frequency-specific AIS distribution in NM during development. So we here labeled NM neurons and stained the AIS with a pan-NAV antibody. And you can see the AIS was very long immediately after the hearing onset, while the length didn't differ between the tonotopic regions. But the length decreased gradually with maturation to a larger extent in high-frequency cells, making the tonotopic difference of AIS length. Importantly, however, when we deprived afferent input during development, it reduced the AIS shortening in high-frequency cells and disrupted the tonotopic difference of AIS length, indicating the AIS plasticity contributes to the fine adjustment of AIS length in individual neurons. We also examined the effects of AIS shortening on the periodic arrangement of axon rings using a super-resolution microscope. These are immunostainings of ankyrin G captured with a state microscope, and you can see periodic signals at an interval of about 190 nanometers, while these signals didn't differ among ages and tonotopic regions. This would imply that the AIS shortening occurred via reorganization of cytoskeletons at distal AIS region rather than compression of periodic structures along the entire AIS. Importantly, it is known that abnormalities of AIS structures are associated with several neurological and psychiatric disorders, such as autistic syndrome, ischemic or traumatic brain injuries, and Alzheimer's disease. So this may suggest that AIS plasticity might be involved in the pathophysiology or pathogenesis of the diseases. So we have studied the molecular mechanisms of this AIS plasticity using pharmacological screening in slice culture of NM, and this is the main topic of my talk. The slices were prepared from animals around the period of hearing onset and cultured on the mill cell membrane for about 10 days. These are NM neurons visualized by a retrograde tracer. So, and the advantage of this culture is the neurons from different tonotopic areas can be easily separated, and their activity can be controlled independently of afferent input. In this culture, we examined the distribution of AIS in high and low frequency cells at 7 and 10 DIV, corresponding to the period of around Hatch. The position of proximal end didn't differ among the tonotopic regions, while the position of distal end distributed more proximally in high-frequency cells at 10 DIV. This makes the AIS slightly shorter in these cells. More importantly, when blocked the spontaneous activity by DNQX and TTX, it elongated the AIS in high-frequency cells and disrupted the tonotopic difference of AIS length. In addition, when we depolarize the membrane by increasing extracellular potassium concentration to about 10 millimolar, it caused a robust AIS shortening again in high-frequency cells. Thus, the activity-dependent reorganization of the AIS occurred primarily in high-frequency cells and making the tonotopic difference of AIS length, which was similar to the situation in vivo. So we examined the effects of AIS shortening on sodium current in high-frequency cells. The whole cell sodium current was reduced by 40% without changes in the voltage dependence of inactivation. On the other hand, sodium current was not detected in outside patches from the soma. This would suggest that the AIS shortening would decrease the sodium channels in the axon. So we next recorded spontaneous activities under current clamp, and you can see burst-like activities were prominent in control, but not after the high potassium treatment. And this tendency remained even after the blockade of potassium current with dendrotoxin. And this confirmed the AIS shortening reduced the activity level of neurons through the decrease of sodium channels in the axon. Thus, so the cultured NM neurons reproduced most features of AIS plasticity in vivo and should be a good model to explore the molecular mechanism of this plasticity. So the AIS shortening is expected to occur via elevation of intracellular calcium due to activations of glutamate receptors and voltage-gated calcium channels. So we added specific blockers of these molecules in the medium for 3 days during the high potassium treatment and found almost all the blockers occluded the AIS shortening, suggesting that the calcium influx through multiple pathways contribute to this shortening during this condition. So we then explored the intracellular signals of AIS shortening downstream of calcium. So we here added specific kinase inhibitors during the high potassium treatment and found that most of the inhibitors also occluded the AIS shortening. But the result was rather surprising, but we don't think this was an artifact because we kept the concentration of the drug as low as possible, and the activators of some kinases like pKa and pKc caused the opposite effect. Thus, so our results would indicate that multiple signals can trigger the AIS shortening, and there is strong crosstalk among the signals. Importantly, the effects of pKa and pKc activators were occluded by inhibitors of ARC1-2 and CDK5. And ARC1-2 is known as an upstream molecule of CDK5. So we consider that CDK5 contributes to the AIS shortening at the downstream of these kinases. So we tested the contribution of CDK5 to the AIS shortening by introducing dominant negative CDK5 plasmid into NM neurons using in ovo electroporation and overexpressing the genes in the culture. Doxycycline was added 1 day before the high potassium treatment, and it suppressed the AIS shortening in the electroporated site. In addition, on the other hand, the overexpression of CDK5 itself didn't affect the AIS length. while the overexpression of p35, which is an activator of CDK5, shortened the ARS. These results were compatible with an idea that the activity of CDK5 is important for the ARS shortening because CDK5 is ubiquitously expressed and its activity is regulated by the level of p35. So this is a summary of down or up regulation of CDK5 activity. Interestingly, the overexpression of both CDK5 and p35 almost eliminated the ARS. And this might be an extreme form of ARS shortening due to strong CDK5 activity. Then, how does the activation of CDK5 cause the ARS shortening? So it is known ankyrin G is required for formation and maintenance of ARS structure, and it is stabilized by multiple mechanisms, including interactions with microtubules and the extracellular matrix with these molecules, as well as direct binding to actin spectrin meshwork. Among them, we are focusing on microtubules as a target of CDK5. This is related to the fact that microtubules have polarized structure and grow and shrink predominantly from one of its end plus end by regulating polymerization and depolymerization, which is known as dynamic instability. In addition, microtubules have a uniform orientation in the axon with their plus end facing toward distal side. And this is rather preferable in explaining the reorganization of distal ARS structure during the ARS shortening. So we examined the contributions of microtubules to the ARS shortening by incubating the cultures with microtubule stabilizing agent like Taxol and tacrolimus AJ during the hypotassium treatment. And these stabilizers occluded the ARS shortening. Taxol also occluded the ARS shortening during the overexpression of p35. This supports the involvement of microtubule dynamics in the ARS shortening. Then how does CDK5 mediate this microtubule reorganization during at the distal ARS region? CDK5 is known to phosphorylate p35 at T138, and this inhibits their binding to microtubules and promotes microtubule depolymerization. On the other hand, T138 is dephosphorylated by phosphatases like PP1 and PP2A, and this reverses the effects and promotes microtubule polymerization. So we tested the effects of phosphatase inhibitor ocalic acid on the ARS length, and facilitation of p35 phosphorylation by this ocalic acid shortened the ARS, and it was occluded by Taxol. And so overexpression of p35 at the T138, which is a phosphorylation delta mutant, rather elongated the ARS. So these clearly revealed the importance of p35 phosphorylation in the ARS shortening. So this is a current model of ARS shortening. So elevation of intracellular calcium concentration by afferent input activates CDK5 and phosphorylates p35, which facilitates depolymerization of microtubules and destabilization of ankyrin G at distal ARS region, leading to the ARS shortening. So this is a summary of my talk. So clustering of NAV channels at the ARS plays a critical role in tuning and maintenance of neural circuits, and phosphorylation of p35 at T138 controls this clustering by regulating dynamics of microtubules. Thus, so phosphorylation of p35 could be a potential therapeutic target for the diseases which are related to abnormalities in membrane excitability or the AIS. These are my collaborators. I thank them for their contributions. Thank you for attention.
I'm interested in whether you ever looked at calcium release from intracellular stores, because that would also elevate intracellular calcium.
Yeah, that's very important. And so there is a ER-like structure in the axon-ischial segment. It is called the cesternal organelle. And we observed the positive effects by activation of G protein coupled receptors. And it is known that such kind of receptors also exist at the region. So it is very possible.
And I can suggest that you could use very low levels of capsagargan to cause a chronic release of calcium. And that might be another way to elevate your intracellular calcium in your studies.
Yeah, thank you.
Yeah, beautiful work. I have two questions. The first one is that, as you know, there are probably very different various mechanisms of homeostatic plasticity of the AIS. So you think that this pathway of CDK5 might hold for these auditory neurons? Because you know, we found, for example, in hippocampal neuron that CK2 might be involved, and the dynamics of the microtubules might be also affected, as the Matthew group found that the molecular motor, the myosin 2 ATPase, may be also involved in the, in the plasticity of the microtubule dynamics. And the second question, is this mechanism involved in the low and high frequency tuning neurons, or is it something specific to one of these two types?
So for the first question, the CDK5 is working in also other neurons, but the effect seems to be diverse among neurons. Probably one possible explanation could be the phosphorylation state of p35, because the phosphorylation can promote depolymerization at T138, but dephosphorylated state called the opposite effect. So that would be the reason, could be one mechanism to explain the diversity. And other kinases, probably multiple kinases would contribute to this process, and probably they're redundant. And so this might be a safe system to call the homeostatic plasticity. So yeah, it is important to find other pathways in this scheme, and how they interact is a very important issue. And the second question, so high and low frequency, this is very interesting. And this diversity of plasticity is observed in many places. So in our system, the low frequency cells doesn't show any change in the AS length in response to this stimulation. And we think the condition of microtubules is one reason or mechanism. So the microtubule associated proteins, so the stability might be, the condition of the stability might be different. So in cortical neurons, so layer 5 neurons shows very profound plasticity, but layer 2, 3 doesn't show. So that may be related. So it is our hypothesis.
Thank you. Nice talk. I was wondering up here, so these neurons can modify their excitability by changing the length of their AIS, but can they also modify excitability simply by changing the motif, like the composition of different channels within a certain, like increasing sodium or decreasing KSNQ?
Yeah, that's a very important point, because the structure as well as the properties of ion channels is important to determine the excitability of neurons. And we previously reported that the composition of ion channels, particularly the potassium channels, switch from low voltage and strong activated KB1 to KB7, so which further facilitates the excitability during the elongation. When the activity is decreased, then the AS elongate. At the same time, KB1 switch to KB7, then activity is very strongly facilitated. So combination of the structural and biophysical interaction is very important, I think.
So we should thank our speaker.